We also have seen in this trial other disability-related endpoints showing a statistically significant effect. And this was something like a 24-week confirmed disability improvement, where we really have shown that there was a 2.8 times increase in the improvement events for active treatment versus placebo.
Hello, you're watching Proactive. Joining me is Immunic Chief Medical Officer, Dr. Andreas Muehler. Andreas, very good to speak with you. One of the highlights of Immunic's presence at ECTRIMS was the oral presentation and the late-breaking poster on your phase 2 CALLIPER trial results in progressive MS, which was even selected for the Best of ECTRIMS 2025 slide deck. Could you explain why these results are so important?
Absolutely, Stephen. So there are very few successful studies in progressive MS populations, and Calibr really showed a medically relevant signal for disability in both populations of progressive MS, non-active SPMS and PPMS, so secondary and primary progressive populations. So most of the medications, or almost all of the medications that are approved and available for treatment MS, are anti-inflammatory drugs. And when they are applied to progressive MS, their efficacy is usually driven by the remaining inflammatory component that may be present in a portion of the progressive MS patients. But what we have seen here is that our caliper results in disability were not driven by these inflammatory effects. Usually we define it as the absence of gadolinium enhancing lesions, or the presence of gadolinium enhancing lesions show you whether this patient still has an inflammatory component. And when we look at our data, the overall population and then the population without the presence of the gadolinium enhancing lesions, we really see no difference. And to my mind, from what I remember, really the first trial where this is truly the case, that the inflammatory components don't play a role in the disability endpoints. And so Calliper, while it was not powered for these disability outcomes, it's our very consistent signal in disability in terms of different endpoints, different populations, and subgroups. So it was a very consistent signal in this trial. And that's really, I think, supports the hypothesis that there are clinical neuroprotective effects that arise from our mechanism of action of Nurr1 activation. And Vidofludimus calcium is the only and the first medication that is working based on this mechanism of action of Nurr1 activation, which is a known neuroprotective target. We also have seen in this trial other disability-related endpoints showing a statistically significant effect. And this was something like a 24-week confirmed disability improvement, where we really have shown that there was a 2.8 times increase in the improvement events for active treatment versus placebo. And that's a very good value. It was also statistically significant. And on top of it all, the drug has been shown a very good safety and tolerability profile, and it confirmed the very good safety and tolerability profile from clinical trials, and there were no new safety signals. So we believe that both as a totality, Vidofludimus calcium is very well suited to be a drug that will be further explored in progressive MS patients.
Beyond the CALLIPER data, Immunic also presented 3 other posters at the conference, Andreas. Could you give us an overview of those highlights?
Yeah, absolutely. I think we had a very active ECTRIMS meeting. We had 2 posters that were looking at follow-up long-term extension data from our phase 2 trial in relapsing remitting MS. We had 1 poster that looked at confirmed disability worsening events in this long-term follow-up study. And it shows that after 144 weeks of treatment, that's a little bit more than 3 years, more than 90% of patients are still free of disability worsening and basically did not see a substantial worsening of their disability. And that is really, I think, very valuable to see that this effect on disability can be maintained for such a long period of time. And then we had a second poster from the same basically long-term extension study that dealt with the safety and tolerability. And we basically could show that the really serious adverse events in the renal system, in the liver, um, really have very low probability to happen, and it basically maintained the very favorable safety tolerability profile over a long period of time in this study. And then at last, we also had a poster that dealt with some new preclinical data. We really, I think, still working very heavily on and investing heavily in preclinical studies that highlight how the Nurr1 activation really leads to these neuroprotective effects of Vidofludimus calcium. And we presented data from several in vivo models of multiple sclerosis at the ECTRIMS in this poster.
Looking at the broader ECTRIMS conference, what were some of the most discussed scientific and clinical trends this year, Andreas, and how do they relate to the direction MS research is taking?
Yeah, I think this was a very interesting meeting from some of the presentations. I think there's still a lot of new clinical study data and a lot of activity in anti-CD20 therapies. That there are several now on the market, and there is a lot of interest by the MS community, and it's also, I think, the most widely used category of MS treatments right now. And there were new data basically showing more data when and how to apply them to MS patients. And this was, I think, very welcome to the MS community in general. There's also more and more data that shows that these therapies, while they're usually very safe and very tolerable, over time with longer treatment in some of the patients, not in many, but in some of the patients, they tend to increase the number of severe infections and serious infections. And that for those patients, we need a solution. What do we do with these patients if they can no longer extend this treatment with anti-CD20 therapy? And I think there were several new ideas how to deal with this situation. But in general, also, I think for us, that's very important to see this tendency of thinking about sequencing other therapies after the anti-CD20 data. And that's something I think that the safety and tolerability profile and the mechanism of action of vidofludimus calcium, I think we would be well suited for this type of sequencing. So we look at this data and really see that it could be of importance to vidofludimus calcium as well. There were several posters that dealt with more detailed analysis of one of the BTK trials, the Hercules trial in tolebrutinib, and giving us more insights about the subgroup analyses and the more endpoint analyses for this trial, because the whole MS community, this was last year's ECTRIMS meeting, really was very interested in this new mechanism of action. I think one of the highlights to me is that in this Hercules trial, now we could see the data that the disability results were in part driven by the inflammatory component, and those patients who did not have gadolinium enhancing lesions did not have necessarily the same benefit as the overall population. So that was interesting to see for us as well.
I mean, finally, moving on from ECTRIMS and looking ahead, what's the next key milestones for vidofludimus calcium in MS?
Of course, we had the Calibr readout this year that was really exciting to us, and we still have to deal, I think, with some of the regulatory interactions in terms of looking to design a phase 3 trial and need agreement on a potential phase 3 trial for vidofludimus calcium and progressive MS. But in general, the whole company and most of us are looking forward to our readout next year in the phase 3 studies in relapsing MS. We have two ongoing phase 3 trials. We call them ENSURE-1 and ENSURE-2. They're fully enrolled already. We're in the follow-up period, double-blind follow-up period. And both of the trials are scheduled to reveal their data by the end of 2026. And that's really, I think, the biggest milestone that this company, Immunic, ever had in their lifetime. So I think this is very much the most important next milestone for the company and for the drug and hopefully for MS patients.
I hope you'll continue to keep us updated on progress. Thank you very much for taking the time today.
Thank you very much, Steve.